Okay, Dr. Ozier here with some content on the ABO blood type. Now, when we think about the ABO blood type, we think it's the blood type. It's the only one that we ever really talk about. It's the only one I learned about in nursing school, certainly. But it's just one of many, and that's because our blood type is typically referring to the types of antigens that are produced on the surfaces of our blood cells, specifically our red blood cells. And in this one, they're A and B antigens. But there are other antigens that can exist, and those will be different blood types. Let's take this opportunity to recap some fundamentals about genetics, because we all know that the world is woefully confused about some core concepts. And this is a genetics class after all. So you should know that the ABO blood type is caused by the ABO gene, and that this gene exists in a particular location in the genome. We know that genes don't move around unless something happens, like a translocation, so this gene is normally found in one particular place. You don't need to know where that is. It's listed here because, teaching. It's chromosome 9 on the long part of the chromosome, the Q arm, at this region 34.1 to 34.2. Always there, so you need to know it's in one place. You should also know that every single person has 2 copies of chromosome 9, and thus has 2 copies of the ABO gene. We got those from 2 places, and we know what those are. They're the egg and the sperm, each of which delivered a chromosome 9. Thanks, Mom and Dad. And the reason that we know that we all have 2 and not some other number is that there are only a few chromosomes that you can have the wrong number of and still be a living, breathing human. Those are the X chromosome. You can lose one of those. That's Turner syndrome. And then you can get an extra copy of 5 different chromosomes and still be alive. Those are X, Y, 14, 18, and 21, I believe, or 13, 18, and 21. You don't need to know the numbers, but note that 9 is not on the list. Therefore, if you are alive, you have 2 9s, one from Mom, one from Dad. Also, remember that for each gene, we might have different versions of the gene that are possible. In Mendel's pea plants, the versions, which we now refer to as alleles, could be associated with being yellow or green, round or wrinkly. In humans, with the ABO gene, the alleles are A, B, and O, and those are associated with the antigens that you make, as well as other things. We'll get to that in a second. The ABO gene codes for an enzyme, and that all you need to know for our intents and purposes is that that enzyme affects which antigens get produced by our body and attach to our red blood cells. And then, because we know that antigens and antibodies go together like peanut butter and jelly, or literally pieces of a puzzle that fit together, like a lock and key, that you will make antibodies to the antigens that you don't make in your own body. So if you are making A antigens on your red blood cells, in your plasma there will be B antibodies. And were B blood to appear in your body, it would be recognized as foreign by these antibodies. So A blood type makes A antigens. They're depicted here with this roundish shape, and your body would not want to attack any A antigens, but it would want to attack B antigens, and so you would make B antibodies. People who have blood type B, depicted here in the second column, make B antigens on the red blood cells, depicted here with sort of a diamond shape, and they would have plasma cells that have antibodies against the A antigen, because they don't make any A antigen. And so notice here that the anti-B antibody shape is triangular in its receptor, which would bind to this diamond-shaped antigen, and the anti-A has a round end, which would bind to this round A antigen. Okay. So that's the A and B blood groups. If you make both an A and a B antigen with your A and B alleles, you will have an AB blood type. And you will not want to make any antibodies against either A or B antigens, because then you'd recognize yourself as foreign. That would be bad. Bodies are too smart for that, usually. Finally, if you have the O blood group, you make neither A nor B antigens, and thus you make both A and B antibodies, because you make antibodies to the antigens that you don't have. If you don't have either A or B, both would be recognized as foreign by your body. What I want to point out here is that you still have this little black stick that the antigens would attach to, were they present, had you any A or B alleles, which you do not if you are in this O group. There's also an exception to that, we'll get to that later. So you make antigens on your red blood cells, antibodies on your plasma, and those antibodies correspond to the antigens that your red blood cells do not have. Okay. We know that there are genotypes that can reliably predict the phenotype that a person has, and those genotypes depend on which two alleles you have. There are 3 options for alleles in the human population, A, B, and O, and if you combine those into groups of 2, using all those permutations and combinations that you didn't pay attention to in math class if you were me, you would get 6 options. You can have 2 A's, and it's sometimes written like this due to the complexities of blood typing and the idiosyncratic nature of scientists. You can have 2 A's, you're homozygous for the A allele, and you make A antigens with each copy, thus giving you an A blood type. There's a second way you can get an A blood type, and that's to have 1 copy of the A allele, making A antigens, and a second copy, which is O, which produces neither A nor B. Likewise, there's 2 ways you can get a B blood type or phenotype. That's having 2 copies of B, being homozygous for B, or being heterozygous, with that second copy being an O. Notice that if you have a heterozygous genotype, but it's 1A and 1B, you're going to have the AB phenotype, AB blood type, and this is because A and B are what's referred to as codominant. Neither one overshadows the other one. You're going to equally see the expression of both, like we did in this slide, where we've got both A and B antigens on the surface of our red blood cell. Finally, you can have the O blood type with neither A nor B antigens attached to our red blood cells, and you need 2 O alleles to get that combination, that blood type. There is some evidence that blood type is linked to some health outcomes of interest. I stumbled across this paper while preparing this lecture. I know that there's a lot of woo out there surrounding like, oh, if you have this blood type, you should eat this diet. We are not going to go there. I think there's a lot of missing information to really understand that, but there has been some compelling evidence in cancer studies that show that your blood type can predict whether or not you survive from cancer, which, if you're going to get cancer, in my opinion, you should survive. That would be the ideal. That would be my goal. Unfortunately for me, I'm not an A blood type, so I'm not at increased chance of surviving ovarian cancer, which is what this study was of. So there are 3 panels to this graph. The first one looks at survival based on blood type, and the second and third look at survival based off of a particular allele in the ABO genotype. So on the x-axis, you see how long people are surviving with a maximum study period of 10 years in this instance, and on the y-axis, we see the percent of people who are still alive. So at the time of diagnosis, time 0, 100% of the people in the study were alive, obviously, because they consented to be in the study and all that. After that, people started dying. And you can see that by the time you get out to 10 years, people who have the A blood type were still about, I don't know, maybe 40% chance of being alive out to 10 years versus like 25% for people who had a different blood type than the A blood type. That's a pretty big difference. Then they broke it down by genotype, and so for this particular allele, you can have a C or a T. So you can be a CT heterozygote, a TT homozygote, or a CC homozygote. And basically the homozygous type A associated genotype here was associated with improved survival. However, the second allele made a bigger difference. How do we tell that? Well, at 10 years at least. At 10 years, there's a bigger gap between the two survivals. I wish they would have repeated the y-axis so it was easier to interpret this, but anyway. So evidence here that your blood type, aka your phenotype, as well as specific genotypes, predicts how well somebody will survive from ovarian cancer. Codominance is the joint expression of two alleles as opposed to the suppression of one by another. If we think about Mendel's pea plants, there are yellow and green peas, and tall and short peas. Tall peas are dominant to short peas, so if a pea plant is heterozygous with one tall and one short allele, it will be tall. Tall trumps short. You don't get a mixture. It's not like a medium-sized pea plant. In the case of codominance, you're going to see both. So picture a flower where you can have a white allele or a red allele. If red is dominant to white, having one red and one white would make you red. Likewise, having two red alleles would make you red. You would need to get two white ones to be white. In the instance of codominance, if you had one white allele and one red allele, you would be both red and white. It wouldn't be pink, it wouldn't be red, it would be like both would be shown. So in the case of codominance with blood type, if you make A antigens and B antigens, they both get attached to your red blood cells. They're both visible and detectable. You express both. You're not trumping one with the other. You don't need two copies of one to show it. Both will be displayed with a single copy in the presence of both. Okay. So if you've got homozygous parents, one who's got an A blood type and one who's got a B blood type, so two copies of the A allele, two copies of the B allele, we know that, so this is supposed to be mom, she's making eggs. The only types of eggs she can make are A allele containing eggs. Dad can only make B allele containing sperm because he has two copies of the B allele, and all the kids will have one B and one A allele. Thus, they will have the AB phenotype or AB blood group because it is codominant. Both will be expressed. In this example, we have a mother who has the A blood group, but she only has one copy of the A allele. The second one would be the O allele, essentially, if we use the other nomenclature. And this one is the B allele from dad, and he's got an O as well. So let's work through this together. I'm going to copy and paste because I'm lazy at typing. So mom is going to give an IA to this kid, but an I to this kid. And dad is going to give an IB to this kid, and this kid. Mom would also give her IA to this kid, but dad would give an I And then finally, mom would give an I to this kid. So this is the O allele. And dad would also give an O allele. So we've got one kid who is AB, has one copy of each allele. This kid would have one copy of the A allele and then an O allele, making them type A blood. This kid would have one O allele and one B allele, making it type B blood. And this kid has two O alleles, making it type O blood. So here we've got an A mom, a B dad, but an O child. This is because each of these parents is a heterozygote, and they have one copy of each allele, and they can pass on this O allele, which is recessive to both A and B. So that's something that I didn't explicitly state. So A and B are dominant to O. It does say it on this slide. So if you have an O and an A, you're going to be A. If you have an O and a B, you're going to be B. And you need two O's to get the O blood group. Okay. Another case here where we've got two parents, each with an A and a B. Oop, don't know why this is animated. Sorry about that. So mom is going to give an IA here and here. She'll give her IB here and here. Dad's going to give his A here and here. And the B here and here. So if you have two heterozygous parents who have both of these codominant alleles, A and B, there's a chance that the kid will get an A from both parents and B, an A blood type homozygous individual. There's a chance that the kid will get an A and a B, one from each parent. That'll happen 2 out of 4 times, and they would be an AB blood type. And then 1 out of 4 chance of having 2 B alleles and a B blood type. So can 2 AB parents have a child who is type O? According to this pedigree, the answer is no, and typically the answer is no, but there is an exception to this rule that is not non-paternity. So sometimes it could be an IA, well, no, an AB mom couldn't have an O kid, even if she had cheated on dad with another person, because the kid's going to get an A or a B from mom. If she was heterozygous and had one O allele, the kid could be O if she cheated on an AB dad. So we'll talk more about this in a second. This comes back to these little stalks. So back in time here, this stalk on the blood cell that the antigens attach to once they are made. This is called substance H. And you need it to attach the antigens, and there is a certain recessive genotype that gives you a phenotype where you lack substance H. In this condition, people can produce A and or B antigens, but not have them attached to their red blood cells because they lack the stalk that connects them. And this is independent of your ABO blood type because it's on a totally different chromosome, because genes exist at a particular location. ABO is on one chromosome, this one is on a different chromosome. And so this is an example of a genetic phenomenon called epistasis. This is an exception to Mendelian inheritance. It is where we have the suppression of the phenotype associated with one gene by another phenotype associated with another gene. So in this case, having the little h genotype gives you no substance H, and it doesn't matter how many A or B antigens you make, you're not going to be able to attach them to the stalks that don't exist. So it's trumping the effect of the other gene locus. So here we see, an AB mother, so that's like similar to our last Punnett square, but now dad is A only, so he's got one O allele. But non-maternity is not really a thing, so it's not like baby switched at birth, maybe. But for the most part, this kid would need to get a second O allele from its mother, her mother, to manifest with the O blood type. Mom would have given either an A or a B, but this individual could have developed their O blood type by lacking substance H, which is the Bombay phenotype. So a quarter of the time, two heterozygote parents will have a homozygous recessive individual that does not produce substance H and will be type O by default. And this is called epistasis because your H substance gene trumps your ABO gene if you don't have H substance.